Alright, so now we will start discussing the tertiary structure of proteins. Now before we begin, let's refresh your mind. Remember that primary structures are really made up of a series of peptide bonds. Now secondary structures, be it alpha helices or beta sheets, are connected via hydrogen bonds. So primary structures beta bonds, secondary structures hydrogen bonds. Like, what sort of bonds are found in tertiary structures? And before we discuss that, let's zoom in on this secondary structure over here. Now, by the way, more than two secondary structures can be connected with each other. So you have this beta sheet over here, and you have an alpha helix over here, and they're connected with each other. And obviously this is not a beta turn, this is a loop. Loops have the ability to connect different secondary structures. And now just because you added two secondary structures together via bonds, right, in this case, via loop, does not mean that you have reached another degree of protein structure. This whole thing is still called a secondary structure, right? And as I said, it's not a different degree of structure just because you've added them together. They're still called secondary structure. And you will understand this more as we discuss the reason behind the process of forming tertiary structure. So as we said, let's zoom in on this region over here, this region of the secondary structure. There you go. Now, in alpha helices, I forgot to mention that R groups, they tend to face the external. So you see this R group, it's facing the external, it's facing outwards. So it's not here, no, it's facing outwards. And you can see this clearly over here. Now this region of the alpha helix, you have the R groups facing outwards. Now these R groups are going to form bonds with each other, right? Now what kind of bonds? We're going to that shortly. But when you have the bond formation between R groups, this whole thing will then, after the binding of R groups together, this whole thing is going to fold onto itself. Now when it folds onto itself, it forms a three-dimensional structure. Now that three-dimensional structure is what we call a tertiary structure of protein. So primary structures and secondary structures are two-dimensional. Tertiary structures are three-dimensional, right? And this three-dimensional structure is achieved by interactions between R groups, right? So R groups interact with each other. They allow this whole thing over here to fold onto itself and form a three-dimensional structure called the tertiary structure. So primary structures have peptide bonds, secondary structures have hydrogen bonds, tertiary structures have bonds between R groups. Now what sort of bonds exactly? Well, the first type of bond you can get between R groups is a disulfide bond. So you have this chain of amino acid and this chain of amino acid. Now they exist in a secondary structure for now. Now by coincidence, you have two cysteines who are adjacent, that are adjacent to each other. Now these cysteines, they're basically the same amino acids, so they'll have the same R group, CH2 and SH. Now what is going to happen between these two cysteines? These two hydrogens will be removed, and these two sulfurs will form a bond as such, right? So again, you remove the hydrogen from here, now these two sulfurs can connect as such. This connection is what we call a disulfide bond. Now when you have two cysteines, notice the E, when they connect with each other, they form one molecule called a cysteine. So two cysteines form one cysteine. And cysteine, as we said, is when the sulfur from the cysteine binds to the sulfur from another cysteine. Now, remember, this thing over here is the R group of both respective cysteines. And as we said, when R groups interact, they form what? They form a three-dimensional structure. They will allow this whole thing to fold and form a three-dimensional structure. And this three-dimensional structure is what we call a tertiary structure. Now, by the way, disulfide bonds only exist between cysteines. So this is the first type of bond that can exist between R groups. Another type of bond that can exist between R groups is something called ionic interaction. So notice aspartate over here and lysine over here. Now, aspartate has a carboxylate in its R group. Now, this is totally different from the carboxylate which is bound here. This is totally different. This is a carboxylate that happens to be in the R group of aspartate, and it's negatively charged. Lysine has NH3 in its R group, and it's positively charged. Now, NH3 and COO negative, right, will form a bond between each other. Now, this bond is called an ionic interaction. So you have one negative molecule and one positive molecule. They interact with each other to form an ionic interaction. Now, obviously, these two things, right, are found in the R groups, and so when they interact, they're basically forming interactions between two R groups. This interaction will allow this whole thing to fold onto itself and form a three-dimensional structure. And by the way, the positive here will cancel the negative here, and so the net charge will be neutral. So this is a second type of bond that can exist between R groups in order to form a tertiary structure, the three-dimensional structure. Now, another type of bond that can exist between two R groups is what we call a hydrophobic interaction. This is not a bond, this is an interaction. What kind of interaction? A hydrophobic interaction. So you have leucine over here and alanine over here. If you remember, these two amino acids are hydrophobic amino acids, right? Now, it happens to be that when two hydrophobic amino acids come together, they form this sort of invisible interaction between them. There's some interaction between them over here. There's no bond, there's no tangible bond between them. There's sort of an invisible interaction between the R groups of leucine and alanine, which are both hydrophobic amino acids. So this interaction between two hydrophobic R groups is called a hydrophobic interaction, and it also allows for the folding of all this thing to form what we call a tertiary structure, the three-dimensional structure. The last type of bond that can exist between two R groups is called a hydrogen bond. You have serine over here, glutamine over here. Glutamine has oxygen in its R group. Of course, amongst other things, serine has hydrogen in its R group, amongst other things. So the hydrogen from serine will bind to the oxygen from glutamine. This bond is called a hydrogen bond. And this is obviously different from the hydrogen. It's the same hydrogen bond as that exists in primary or secondary structure, but this hydrogen bond is different in that it exists between two R groups. This is still a bond between two R groups, because this yellow thing over here is an R group, this yellow thing here is an R group, and these two yellow things, they interact with each other via hydrogen bonds. And so this allows this whole structure over here to fold and form a three-dimensional structure, which we call the tertiary structure of a protein. So let's recap. A tertiary structure is when two R groups of different amino acids, they form a bond or an interaction between each other. This will allow the whole secondary structure to fold onto itself. Now, once it's folded onto itself, it forms a three-dimensional structure. This three-dimensional structure is called the tertiary structure of a protein. Now, this folding process we've mentioned is aided by this protein complex we call chaperones, right? Now, obviously, this is a secondary structure. It enters inside the chaperone, and with the help of ATP, the secondary structure undergoes the process of folding, and then it's released, right? and this is aided by what we call chaperones. Now, malfunctioning of chaperones might result in misfolding of an aggregation of proteins. So chaperones, they play a key role in preventing misfolding and aggregation of proteins. Now we move on to the quaternary structure. Now, not all proteins will form a quaternary structure. Most proteins will stop at the tertiary structure, but some proteins like hemoglobin will advance to form a quaternary structure. Now, what is a quaternary structure? Now, most textbooks will say that a quaternary structure is two or more polypeptide chain. I find that a bit confusing because beta sheets, which is a type of secondary structure, is made of more than one peptide chain. Now, beta sheets are not made of more than one polypeptide chain, it's made of more than one peptide chain. And obviously, I think you're a bit confused with this. It's okay, I have a better explanation of what a quaternary structure is. So a quaternary structure is basically a structure of a protein that has two or more tertiary structures which are connected. So in the case of hemoglobin, you have this tertiary structure over here, you have this tertiary structure over here, you have this tertiary structure at the back, you have another tertiary structure at the back. Notice, by the way, how this is three-dimensional. This thing over here, for instance, is three-dimensional. That is something we've said regarding tertiary structures. They are three-dimensional. Now, getting back to hemoglobin, these four tertiary structures will bind to each other via one of these bonds: hydrogen bonds, ionic bonds, hydrophobic interactions. So this tertiary structure might connect with this tertiary structure via one of these bonds or one of these interactions, right? So a quaternary structure is when two or more tertiary structures connect with each other via one of these interactions. That is a quaternary structure. And as we said, not all proteins will form quaternary structure, only some, like hemoglobin. That was the end of the protein structure, the normal mechanism of protein structural formation. The next video we will be touching upon the clinical aspects of improper protein structural formation and protein aggregation. If you have any questions, please don't hesitate to write them in the comments below. I will respond.